Hello and welcome to Astrobiology Podcasting. In this lecture, we are going to do a mini science seminar here for our scientific inquiry activity. And I'm going to tell you a little bit about some of the research that my lab does on ecosystems called microbialites. And again, I want you to think about those questions we talked about in the scientific inquiry lesson as I am talking about the science, and try to poke holes in what I'm trying to say and see what you can come up with in terms of questions for the scientific inquiry exercise. So what I work on are what are called microbialites. These are ecosystems that have been around since the beginning of life on Earth. And I'm specifically going to talk about something called a thrombolite. And a year in the life of the thrombolite is a study that we did over the last few years where we started to look at these thrombolites. Here's an example. They kind of look like rocks, but they're actually living ecosystems found in the intertidal regions of some tropical regions. And what we're trying to do is understand how are the microbes coordinating their activities and existing within these communities, and how are these microbial processes changing over both diel throughout the day and seasonal cycles. And these are just some other pictures. We are working in the Bahamas, specifically in an island called Highborn Key in the Bahamas, because they're relatively close to Florida. So before I go much further, let's define what I'm talking about. What is a thrombolite? I know it sounds something you might order in an Italian restaurant, but it's actually what's called a microbialite. And microbialites are sedimentary structures. What that means is these are deposits of calcium carbonate made by the activity of microbes. And they're accreted through both the interactions and the processes of many different microbes. And we'll talk a little bit more about that. And there's many kinds of microbialites. So microbialite is kind of like an umbrella term. And thrombolites are the one that I'm going to talk about now. And these have what are called clotted appearances. They don't have really distinct geological structures. They just kind of look like these buildup of calcium carbonates. And here's a picture of them. They tend to occur in the upper intertidal regions, and they tend to get exposed a lot during the normal day. Stromatolites are the ones that you might have heard of before. These are the oldest known ecosystems that we have fossils for today on the modern Earth. And these have it's hard to see, but these have a laminated structure. They're deposited in layers by the microbial communities. We call it a laminated internal organization. And stromatolites, again, they're very ancient systems. And there's another type of microbialite that I don't work on, but they kind of form rings like around a tree, and they're kind of called oncolites. And they are a little bit more rare out there in the modern world. So why should you care about thrombolites? Why are these important ecosystems to study? Well, the modern systems, there's living examples all throughout the globe, but they're a little more rare than the stromatolites. But the modern systems can be like a window into the past. They can help us understand if a living ecosystem can survive today, maybe that it was able to survive under similar conditions in the past. So they're what we call an analog into the past. Thrombolites are also tough. They live in tough environments. They can live completely desiccated out of the water. They can live in high temperature, high UV radiations. So they're also great for studying how microbes can respond to environmental stress conditions. Now, I mentioned I work in the Bahamas. The thrombolites of Highborn Key, Bahamas are a great model. They're very easy to get to, as you can see how shallow they are. You don't even have to really get wet when you work on them. And here's a picture of the exumas here. Here's Florida here to give you an example, and a blow up of the island. And the thrombolites all exist along the margin here, the eastern margin of the island. And here's just a close-up picture. If you zoom in onto the surface, you blow it up, you see these nodule-like structures on the surface. So if you cut one of these nodules in half, you can kind of see these little vertical striations. And here's a microscopy image of the same picture. And what you can see are basically bundles of cyanobacteria. And through DNA sequencing, we've actually sequenced the 16S rRNA gene, the ribosomal rRNA gene, of these microbes. We dissected them out and extracted the DNA and did a PCR amplification on it. We were able to identify the species name was Dicothrex. This is a type of cyanobacteria that's very ancient. And we found that these cyanobacteria, through their metabolism photosynthesis, is what's driving calcium carbonate precipitation. What happens is the microbes take up carbon dioxide from the atmosphere through photosynthesis, and what they're doing is they're using up carbon dioxide so fast that it is forming hydroxyl radicals, and this hydroxyl radical actually pushes the pH slightly lower, and that triggers calcium carbonate precipitation. And where you see arrows here, we've actually extracted out the calcium carbonate, and we've actually done some X-ray diffraction analysis, some mineralogy analysis, and found that that is indeed calcium carbonate. So the microbes are actually driving the formation of these structures through carbonate precipitation. Okay, so we knew that the cyanobacteria here were taking carbon dioxide and forming these structures, because you can see the structures there. But how were they doing this? What are the biological mechanisms that they were doing it? And also, we know that the cyanobacteria were the dominant microbes in this community, but were there other microbes, and were they alone? And did they also help make this structure? And so one of the first things we wanted to know is, what the heck are they doing? How are they doing this carbon precipitation activity? I mean, this could be very important for microbes to be able to have this ability to sequester and remove carbon dioxide from the atmosphere. So what metabolisms are present, and are they active within the microbialites? So what we did is we targeted the metagenome. The metagenome is basically all the genomes of all the microbes found within the community. It's all the genes that they have. And then we targeted the metatranscriptome. These are not just the genes that they have, but this is the genes that they're actually using. So we sequenced both the metagenome and the metatranscriptome, and we did this again at 4 time points throughout the day over several different seasons. So what we did is, here's just a snapshot of some of the first data we did. And the different colors represent all these different categories of functional genes that we found. The outer ring is actually the metagenome. And this was, again, we just captured the entire surface. So we took the first top 1 centimeter of the microbial community, we mixed it all up together, and then we sequenced everything all together. When we did the metatranscriptome, we did things a little differently. We actually sectioned them based on the depth profile. We wanted to kind of see also not only a temporal or time changes within the mats, but we also sequenced them according to depth. And so you can just see here, 1, 2, 3, these are basically depth profiles down deeper into the mat. And again, the colors, all the colors, whether it's in the first row, second row, third row, they all correspond to these different functions. But what we clearly saw was in the metagenome, the photosynthesis genes, they only made up a small percentage of the genes contained within the metagenome, but there were a lot of other genes associated with many different metabolisms. In the metatranscriptomics, those are the genes that the organisms are actually using at that moment in time. That was predominantly, at least at the surface, was predominantly photosynthesis. There was also some carbohydrate metabolism. Carbohydrate metabolism is red, and then protein metabolism. They're making lots of proteins as well. But as you go deeper and deeper into the mat, there becomes less and less photosynthesis. And this made sense because as you get deeper into the microbial communities that are making up these thrombolites, it's harder and harder for light to penetrate. So the previous data, this previous slide, really gave us an idea that the metagenome might be really different from the metatranscriptome, meaning just because you have a gene doesn't mean the microbes are using that gene. So what we did is we dug a little further into the metatranscriptome and really wanted to see how these taxa metabolisms varied over this diel. We looked at 6 a.m., 12 p.m., 6 p.m., and midnight, and how did that vary over seasons? And we looked at March, August, and October. And we generated metatranscriptomic libraries. We did everything in duplicate. We did 2 copies of everything. And we used a platform called Illumina Next Seq 500. That's how we generated our DNA sequences. High quality recovered transcripts. And we filtered those using several bioinformatics techniques. And then we recovered 900 million rRNA transcripts. That's the ribosomal RNA gene. And then 300 million message RNA transcripts. We felt that our sequence recovery was very robust, and we didn't do any additional tests to see whether or not we had enough sequencing coverage to capture the community. We felt that 900 million and 300 million was enough. What I wanted to know is, does the seasonal and diel variation occur between all the different libraries? And so we did replicates of all the different 24 libraries that I showed you, and what we do is we plot them on what's called a PCOA plot. And here we can start to get an idea of how similar they are. Are all the replicates that came from the same mat similar, or the same time point similar? Or are they all over the place? Are there any patterns to the data that we can distinguish? Are there any differences? Are there any similarities? So here what I've done is I've plotted the samples from March, August, and October. And what we can see is there is about 11% of the variation here. So there's about 11% difference or variation that we can explain by the seasons. So there seems to be some seasonal variation occurring within these populations of microbes within the thrombolites. So I think that this direction here, PC1, this direction represents seasonality, so that August and October look really similar. I don't think that there's significant differences between the microbial populations between August and October. And as you can see, the temperatures are pretty warm. There's probably not much fluctuation in the environmental variability. But March is colder, and I think that the samples are much more susceptible to cold, and they're expressing different genes, and they're looking a little bit different in March. So one of the first things we did was to look at whether specific microbes were active during different parts of the day. There's many different microbes. Here's just a list of some of the dominant phyla. This is all done at the phyla level, showing you how many microbes are present. It's a really diverse community. And proteobacteria are by far the largest and the most diverse. So even though the Dicothrix was the biggest microbe, it was visibly visible and formed these vertical kind of orientation, these little nobules on the surface of the thrombolites. There were many other microbes present within that community. But cyanobacteria were the second most abundant groups. So then what we wanted to do is look at specific metabolisms. We looked at the taxa and when they were active. What about specific metabolisms? My hypothesis was that it's the cyanobacteria and their photosynthesis that are driving the precipitation. So here what I saw was when I plotted all the photosynthesis genes, I saw a peak at noon and then it would diminish at 12 a.m. This makes sense because at night they can't photosynthesize as actively as they can during the day because they don't have as much light energy. But that was photosystem 2. Interestingly, we didn't see a statistical difference in photosystem 1. So there was definitely a difference between the two photosystems. Photosystem 1 can be used by not just cyanobacteria, but other microbes that can actually do photosynthesis at night. When we looked at other metabolisms, we also found that nitrogen fixation was very abundant. We recovered over 12,000, up to 12,000, I should say, different transcripts. And when I say a transcript, that's an mRNA product. That's an actual copy of that particular gene that was transcribed and used, or is ready to be used by the microbe. And so again, that kind of peaked at around 6 p.m. and was still very high around 12 a.m. That suggested that nitrogen fixation in the thrombolites was happening, but it didn't happen during the day, and it happened at night. The other thing we also did is we looked at dissimilatory sulfate reduction. This is a metabolism that has also been shown to precipitate calcium carbonate. When this metabolism happens, it can actually also cause the pH to become more basic, and that can facilitate calcium carbonate precipitation. So we looked at one gene involved in that process, the DSR gene, the dissimilatory sulfate reduction gene. And although there weren't as many copies as there were in the nitrogen fixation, we did see a diel cycle pattern, although it wasn't statistically significant. So we do see somewhat of a trend, but there was a peak in the August samples here in the blue compared to the other 2 months. But in general, we didn't see a statistically significant trend of this metabolism. There were also other metabolisms like denitrification and methanogenesis that are also involved in carbonate precipitation, but I'm not going to talk about those today. So in summary, when I look at this data set of transcriptional changes, and I actually go through all the different time points, not just the August one that I showed you in the last slide, we really see a highly diverse, transcriptionally active community. They're very dynamic, with most of the cells belonging to the proteobacteria and the cyanobacteria, specifically groups of coccoid cyanobacteria and Rivulaceae. Rivulaceae is the family that Dicothrix belongs to. What we found was the most transcriptional active time period appears to be noon. Then sunrise was the second most abundant. It didn't matter what season it was always the highest amount of activity happened at noon. Nitrogen fixation, denitrification, methanogenesis, all those peaked at night, but they had much lower expression levels than photosynthesis. That to me suggests photosynthesis is the most dominant metabolism in thrombolite communities, and photosynthesis is responsible for most of the carbonate precipitation in this community. Dissimilatory sulfate reduction, we followed that one gene. It had relatively low transcriptional activity, although it was a little bit higher at night, but the low expression levels suggest that that metabolism isn't very important in the thrombolite ecosystem. The next thing we want to do is start building integrative networks where we can start to reconstruct what genes are expressed when, and start to look at the co-expression networks and really how do they regulate these metabolisms over time. So it wouldn't be a science talk if I didn't talk about the people who actually helped do the work. So this is Artemis Louyakis. She was the graduate student that did the work, and these are all my lab members. You can see that water is definitely a theme of my lab here, and I thank NASA Astrobiology for funding this project. Now again, I have misstated things. I have elaborated on things. I've been a little short on things, but hopefully you can now use the information that I've given you to kind of tease apart, ask questions about what I said, and pose a few questions for the scientific inquiry exercise. And I'm happy to talk to you in more detail if you want to learn a little bit more about metagenomics and metatranscriptomics in thrombolites. So thanks for listening.